With the field of medicine rapidly progressing, our capabilities are nearing the realm of science fiction. We have learned to perform highly complex life-saving operations and treat diseases that would have been a certain death sentence 100 years ago. One of the most important advances that have made this possible is a discovery made in 1928, the discovery of penicillin, the first antibiotic. Since then, several others were found or created synthetically, and now, for a modern person, it would be difficult to imagine a world where a simple infection or surgery could be life-threatening. Such a world, however, is once again becoming a possibility. Bacteria are evolving, becoming able to survive treatment with antibiotics, and due to overuse or incorrect use of antibiotics, these resilient pathogens are growing more and more prevalent. Antibiotic resistance is a rapidly growing phenomenon that has become one of the largest global threats according to the World Health Organization. This threat is deadly. Old killers, diseases such as pneumonia and tuberculosis, are returning as drug-resistant superbugs. This threat is omnipresent. E. coli, which are part of our gut flora and responsible for the majority of common bacterial infections, are turning impervious to antibiotics. Unsurprisingly, scientists are locked in an arms race with these evolving pathogens. But discovery or design of new antibiotics is a long and difficult process that can take years, time we do not have. Furthermore, it only postpones the inevitable. Bacteria will eventually evolve to survive these as well. Thus, it is imperative that we find alternative ways to combat bacteria.
Is the enemy of my enemy really my friend? This idea is behind one of the most promising solutions for the growing threat of antibiotic resistance: phagotherapy. Phagotherapy is an approach that utilizes bacteriophages, which are viruses that exclusively infect bacteria, to combat bacterial infections. While it is not yet a universally accepted treatment, it has yielded promising results in laboratory studies and individual cases. There are many types of bacteriophages, with different modes of infections, reproduction strategies, and host specificities. Right now, scientists are working on modifying and adapting phages to more effectively target and kill pathogenic bacteria, especially those that carry antibiotic resistance genes, while being harmless to the patients and the environment. Thus, as with any cutting-edge area of synthetic biology, many past and present IGEM teams have built projects around phagotherapy. We are the Team Paris-Saclay 2020, and our project on gene entanglement is inspired by a phenomenon first discovered on the 5X174 bacteriophage, and that is overlapping genes, where the same structure of DNA encodes two different genes simultaneously. One possibility for overlap is simply one strand of DNA coding for one gene, while the opposite strand of the same stretch of DNA coding for a different gene. However, our team has put its interest in another form of overlap: unidirectional overlap, when both genes run in the same direction but are offset by one or two nucleotides and are thus out of frame. In this case, the same DNA sequence will produce two different proteins depending on the reading frame being translated. Since the initial discovery, many such genes have been found in organisms from viruses to eukaryotes. However, an exciting new development may allow us to harness this phenomenon for synthetic biology. Recently developed constraining adaptive mutations using engineered overlapping sequences, or cameos, software. It allows us to artificially entangle genes, creating combinations of overlapping genes that do not exist in nature. We believe that this technology will have many uses in the field of synthetic biology, from biocontainment to design of more stable genetic constructs, and more specifically in the field of phagotherapy for improving phage design. One possible application is the stabilization of any genes added to the phage. Viruses are known for their fast rate mutation. Genetic entanglement can provide a solution by entangling any new added genes with the ones essential for the phage, such as those involved in phage replication. Those are the most concerned. This way, any mutation in the sequence of a gene of interest also occurs in the essential gene, impeding the ability of the phage to replicate, and the mutation is not passed on. Another potential application is the addition of new properties to phages with small genomes. Phagotherapy often uses large phages that carry many genes. This increases the risk of horizontal gene transfer from the phage to the bacteria, which is a major concern in phagotherapy. We hypothesize it could be possible to create a safe phage by adding genes to smaller non-lytic phages with smaller genomes, such as a filamentous phage. It might even be possible to add a CRISPR-Cas system that would selectively target and kill bacteria with the antibiotic resistance genes. Due to the smaller genomes of these phages, adding multiple genes might destabilize the phage or impair its assembly. Genetic entanglement could be a viable solution for fitting more genes into smaller phages, as more genes can be added without greatly enlarging the genome. Whether reducing the number of mutations or introducing large genes into small genomes, the gene entanglement project has the capacity to push the field of phagotherapy and extend beyond, into fields such as genetic engineering and biocontainment. We are witnessing the first steps of a technology for which we envision a great future. Thank you for listening. We are Team Paris-Saclay and we are going to go and entangle some genes.